Okay, synthetic theory of evolution or the neo-Darwinism or the modern theory of evolution. As per the theory, the synthetic theory of evolution, evolution is based on various evolutionary processes. Okay, these processes include mutation and resultant hereditary variations, natural selection, genetic drift, and now we have the isolation. Okay. Any external or internal barrier that prevents interbreeding between populations can be referred as isolation. And as a result of this, the population of a species is either separated into smaller units or exchange of genes between them is prevented. Okay. So, isolation can result in two processes. One where a population of a species gets separated into smaller subpopulations, and what happens as a result? Between the subpopulations, breeding cannot happen, or it may be prevented, maybe because of some kind of physical factors or some kind of geographical features like rivers or mountains, etc. So in such case, what happens? Subpopulations may not be able to interbreed. So it is one kind of a prevention of interbreeding. Secondly, the populations may be remaining together, but due to certain other reasons, gene exchange or gene flow between them is prevented. So in both the cases, it results in what you call as separate populations or later speciation. So hence, genetic factors such as mutation, recombination, genetic drift, natural selection, etc. occurring in one population will not have any effect on the other population. These subpopulations will be experiencing different kinds of what you call selection pressures, even mutation, recombination, etc. Okay, so in the course of time, each separated population forms a separate unit. Thus, isolation helps in splitting the population of a species into separate units and their subsequent evolution into distinct species. So, isolation can finally result in the formation of different species. That is, isolation is a reason for speciation. Okay. Now, according to Romanus, without isolation or prevention of interbreeding, organic evolution is never possible. Okay. So, isolation or prevention of interbreeding is a must for the organic evolution and speciation to happen. I hope it is clear. Okay, so this is about the isolation. Now, how does the isolation happen? Okay, and that is what is referred as isolating mechanisms. So, we will be learning about the different types of isolating mechanisms. Isolating mechanisms have been variously classified by a number of biologists, like Mayer in 1942, then Huxley in 1942, Stebbins in 1950, Dobzhansky in 1955, and many others. Okay, and here we are going to deal with Dobzhansky's classification. And as per Dobzhansky, the isolating mechanisms can be divided into two major types. Okay, geographic isolation and reproductive isolation. Okay, geographic isolation, the name itself says they are geographic barriers like mountains, land, and water masses. Thick forests, vast deserts, etc. Okay, these prevent two reproductively active populations from coming into contact. So, it is like a population is there and it had got this members of the particular population of a species got separated into two subpopulations, and these two subpopulations are interbreeding, but because of a geographical barrier like desert or water mass, etc., these two subpopulations cannot interbreed. Okay. So, this results in two subpopulations, and these subpopulations, if it is experiencing different kind of selection pressures, slowly, gradually, what happens is it may result in the formation of speciation from these two subgroups. Okay. The next one is the reproductive isolation. Reproductive isolation includes physiological, ecological, mechanical, cytological, genetic barriers, etc. This prevent interspecific mating or reduce the formation of viable zygotes or hybrids. Okay, so there are reproductive isolation will be looking in detail, but this reproductive isolation in general, it prevents interspecific mating. Okay, that is between members of different species. Such kind of mating is prevented, and it reduces the formation of viable zygotes or hybrids. Okay, so we will be seeing it in detail in when we discuss about the two types of reproductive isolation in detail later. Okay, now reproductive isolation may be further classified into prezygotic isolation and postzygotic isolation. And prezygotic isolation includes these many types of isolations. Prezygotic isolation, actually it is prevents fertilization and formation of zygote. It is prezygotic isolation. So this isolation prevents the formation of zygote. Before the zygote formation, isolation actually creates a barrier. Postzygotic isolation is where it the barrier, it actually doesn't have any influence on zygote formation. So zygote is formed, but once the zygote is formed, the barrier may prevent the development of the zygote into a viable offspring, or it may prevent the individual to in reaching the reproducing age, etc. Okay, so we will see geographic isolation here. Geographic isolation, when two populations or two subgroups of one population are separated from interbreeding by some physical barriers, it is referred as geographic isolation. Okay, the barriers may include vast stretches of land or water, mountain ranges, deserts, thick forests, etc. These geographical barriers prevent migration and intermingling of the individuals of two populations, and hence interbreeding and subsequent gene exchange between them are completely prevented. Okay, so that is how the isolation will result in separation of subgroups in the population. Okay, now examples include fauna of Australian region, especially marsupials and monotremes. Then another is fauna of Galapagos Islands, and another specific example is with respect to the two species of Drosophila. Okay, we can start with this one. Actually, Drosophila pseudo obscura, they are found in low altitude areas, that is almost at the sea level. Drosophila is it is Drosophila. Okay, sorry. Right. So Drosophila pseudo obscura, it occurs in low altitude areas, while Drosophila personites, it occurs in high altitude areas. So obviously these two species, even though they are very close, even though they are found in the same area, but because they are found in two different altitudes, they never come together for interbreeding. Okay, so they are always at two different heights. Okay, high altitude means maybe at a higher mountain level, and low altitude area means almost like on the sea, like along the sea level. Okay, so these two may not get a chance to intermix or intermingle, and then giving rise to interbreeding and giving rise to genetic exchange. Okay, so Drosophila pseudo obscura and Drosophila personites, they are from different altitudes but same geographical areas, but because they are from different altitudes, they are separated from interbreeding. Right. Now regarding the fauna of Australian region, geographical isolation on land is provided by the fauna of Australian region. Australia, Africa, South America are all located in the southern hemisphere. They occupy approximately the same range of latitude and have almost the same variety of habitats, yet they have widely different mammalian fauna. In Australia, we can find the marsupials. Marsupials include the pouched mammals, isn't it? Kangaroos, etc. So these marsupials are totally absent from Africa and are represented in South America only by the opossum. Again, with respect to monotremes, we find that spiny anteater and duck-billed platypus, they are found in Australia and nowhere else in the world. But Australia has very few placental mammals, except those that have been introduced by man. So on the other hand, so I hope it is here the geographical isolation is responsible for the differential distribution of the marsupials, monotremes, and even the placental mammals. Okay, then regarding the fauna of Galapagos Islands, very typical, like a very unique faunal distribution can be found in Galapagos Islands. Galapagos Islands is situated on the equator, almost like 900 kilometers west of Ecuador in South America. And here we can see that the Galapagos Islands were formed by volcanic action, and these islands were initially without any life. Okay, but later they were colonized by animals and plants from the mainland, that is the South American land, because that is the closest one, and large bodies of water isolated them from the fauna of the mainland, that is South America. Hence what happened, so some animals and plants colonized from the mainland, right, and these few populations, they survived on the island, but what happened is the ancestral or the parental population in the mainland and the what you call migrated population on the Galapagos Islands, they were completely separated by a vast stretch of water. Hence they evolved along their own lines into separate species. That is, the members on the Galapagos Island experienced different kinds of natural selection pressure, while that on the mainland had experienced different, completely different selection pressure. So what happened is they started to evolve into their own lines into separate species, acquiring adaptive modifications in response to different modes of diet and variety of habitat available to them in the particular island. So thus, though fundamentally similar to the mainland forms, they differ from them in details. And there are about 76 species of land snails, 13 species of Darwin's finches, terrestrial and marine species of giant iguana lizards, marine lizards, giant land tortoises, etc. So when Darwin visited the islands in 1835, he observed variations in the fauna, and also he observed that this could be due to isolation and independent evolution that may have resulted in this particular kind of a unique faunal distribution of on Galapagos Islands. Okay, so in all these cases we can see that large bodies of ocean waters, especially the Australian, faunal composition of Australian region and faunal composition of Galapagos Islands, these two arose because of large bodies of ocean waters that formed effective barrier to land animals. Okay, so even rivers, lakes, ponds, they form effective isolating agencies. So I hope it is clear, right? Geographic isolation. Then we have here a few other examples of geographic isolation. Here you can see two different species of anisotromus. There is a porkfish, okay, anisotromus on the the eastern side of the Isthmus of Panama. Isthmus of Panama is a small land mass which connected South America with North America, and on the eastern side you can see Anisotromus virginicus, and on the western side you can see Anisotromus tenuatus. Okay, these two populations, they can not interbreed because of the geographical barrier. Okay, they may have arose from the same ancestral stock, but because of the geographical barrier they cannot interbreed and cannot result in the formation of viable offsprings. Okay, and finally these are completely different species. Another one is with respect to the squirrel population, the different species of squirrel population on either side of the Colorado River. Okay, the Colorado River, it is Grand Canyon through which the Colorado River flows, and you can see that earlier these squirrel populations had a common ancestor, but as the Grand Canyon increased in depth, the squirrel population got separated into two subpopulations, one on the northern rim and another on the southern rim of the Grand Canyon, and once on the northern rim it developed into kaibab squirrel, and the one on the southern rim it developed into abbott's squirrel. Okay, and these two subpopulations it slowly underwent evolutionary processes, and it resulted in two different species and can now no longer interbreed. Okay, they are completely different species. Okay, but earlier these two species had arose from the same ancestor, and these two could have interbred during that particular time period. Okay, so this is geographical isolation with respect to the two squirrel populations along the two opposite rims on the Grand Canyon. Then another example is with respect to the marine iguana. This we have already mentioned, the faunal composition of Galapagos Islands uniqueness because of the vast stretch of water between the Galapagos Islands and the mainland South America. Okay, and because of that, the populations which migrated from South America to the Galapagos Islands, it started to experience different kinds of environmental conditions, different kinds of selection pressures, and hence it started to evolve into species entirely different from that of the parental population on South American mainland. Okay, so that is the geographical isolation. Now, reproductive isolation. So, what is reproductive isolation? As we have already mentioned, reproductive isolation is one where the there is reproductive barrier. Okay, there is this prevent interspecific mating, or it prevents the formation of viable zygotes or hybrids. Okay, there are two kinds of reproductive isolation as already mentioned. So, prezygotic barrier and postzygotic barrier. Okay, so here you can see the prezygotic barrier acts before the formation of zygote. So, prezygotic barriers include those isolation mechanisms which prevent the formation of zygotes. Okay, postzygotic barrier is those isolating mechanisms which never prevent the formation of zygote, but it actually prevents the formation of viable individuals from the zygote. Zygote is formed, but it may not develop into viable fertile offsprings in the future. Okay, so these are the two kinds of reproductive isolations. First one is prezygotic isolation. Prezygotic isolation, it is a type of isolation that prevents hybridization of members of different populations and thus the formation of hybrid zygotes. So, it is prezygotic isolation referred to the prevention of the formation of hybrids. When is the hybrid formed? Hybrids are formed when there is interbreeding between members of different populations, or we could say different species. Okay, so such kind of hybrid formation is prevented in prezygotic isolation. And there are almost 6 mechanisms of prezygotic isolation. The first one is ecological. Okay, here you can see ecological, or it is otherwise known as habitat isolation or environmental isolation. This occurs when species occupy different habitats in the same general territory. Okay, for example, just imagine two species of beetles. Okay, these two species of beetles, they live on the same wooden log. Okay, I hope you know what is a wooden log. When in forested areas and all, when tree gets older, they fall down, and these fallen tree trunks, that is what is referred as the logs. Okay, and in the tree, in the same log, two species of beetles are found, but one of the species, it lives deep inside the log, while the other species, it usually lives just below the bark. Okay, loosened bark. So what happens is, because of the two habitats occupied by the two beetles, what happens? There is a prevention of interbreeding between them. Okay, so that is what referred to. Same territory, that is a log, different habitats, that is one is deep within, another is the just below the bark. Okay, so there can be plenty of examples here. Here one example is like an insect that inhabits only coniferous forests is said to be environmentally isolated from an insect that inhabits only deciduous trees, even though both live in the same forest area. Okay, I hope it is clear. Now the typical specific example is with regard to the rana species. Okay, the rana, it is a frog, okay, a species of frog, and rana grylleo, it is pig frog, and rana areolata, it is a gopher frog. Okay, this pig frog and the gopher frog, you can see these are two species of the same genus. Then pig frog, they is extremely aquatic and they are found in deep ponds, lakes and marshes, while the gopher frog, it lives in burrows during daytime, but they are active in night and they are found along the margins of swampy areas. And the pig frog breeds in deep water and has no ecological contact with the gopher frog. The reason is the gopher frog breeds in isolated grassy ponds in shallow waters. So the differences in their habitat eliminates possible mating between the two species. Okay. Now there is, even when we speak about the activity, we saw that the gopher frogs are mainly night active, while the pig frogs are day active. So even in that respect they cannot, but here we are speaking about the habitat isolation. Okay, so I hope it is clear. Now the next kind of a prezygotic isolation is temporal isolation or seasonal isolation. Seasonal or temporal isolation occurs when members reach sexual maturity at different seasons of the year. That is, the breeding seasons of the two species may not coincide, although they exist in the same region. Okay. One example is that of, you can see this in among amphibians, many amphibians and insects. Specific examples are here. One is with respect to two species of toads, Bufo americanus and Bufo fowleri, and another is with respect to the two species of Drosophila. Okay. The two species of toads, actually the study was conducted by Blair in 1941 and Corey and Mannion in 1955, and they observed that Bufo americanus, they bred in early rainy season and they preferred forested areas, while Bufo fowleri, they bred in late rainy season. So what happens is, due to the differences in the time period or breeding time, this could never undergo interbreeding. Okay, these two cannot undergo interbreeding. But there are certain times when the two seasons, very occasionally these two seasons overlapped. Okay, in such cases hybrids were formed, but it was very rare. Okay. Another example is that from an insect, Drosophila. Drosophila pseudo-obscura was active in evening, and Drosophila persimilis, it is active in morning. Okay, even though they live in the same area, okay, sympatric, they live in the same area, they breed in the same season, but in different times of the day. Okay, it needn't be in the different season, but even if it is in the same season, if breeding happens during two different times, even that is actually itself prevents the zygote formation, or it acts as a prezygotic isolation. Clear? Okay. So this is about the temporal isolation. The third one is ethological isolation or sexual isolation. In some cases what happens is, breeding is possible but does not occur due to the absence of sexual attraction between females and males of different species. That is, for example, the two species of frog, okay, Hyla versicolor and Hyla femoralis. Interbreeding is possible here, but naturally it never occurs. It is because the two species, the ethological behavior or courtship behavior, all those behavior aspects with respect to the breeding or associated with the breeding are different. Okay. In many species, copulation is actually preceded by elaborate courtship behavior. You may have seen you may have seen that peacocks dancing, isn't it? The male peacocks, they lift up the tail feathers and they show dances. It is actually a courtship behavior to attract the females. Similarly, you may have heard the croaking sound of the frogs. Okay, those croaking sound of the frogs are made by the male frogs to attract the females. So all these behaviors are known as courtship behavior. Okay, and this courtship behavior usually follows the breeding and interbreeding, and then the like mating and all, right. So these behaviors may be different among the species. The very prominent plumage, melodious songs of many birds, the calls of frogs and toads, then the singing, okay, the sound vocalization of crickets and locusts, they are all associated with this courtship behavior. Okay, so differences in this what you call courtship behavior between the species will prevent interbreeding. I hope it is clear. Okay, one typical example is that of the two frog species, Hyla versicolor and Hyla femoralis. Hyla versicolor, actually this study was conducted in New Orleans, USA, and here the closely allied great tree frog and wood tree frog, they frequently breed in the same pond, but interbreeding between them never happened, and it is because the mating calls of Hyla versicolor and the Hyla femoralis are totally different. Hence they are able to distinguish the mating calls of their own species from those of others. That is, the mating call of male Hyla versicolor was different from that of the male of femoralis. Okay, so and it was very easily distinguishable. So obviously they were able to distinguish the mating calls of their own species, and hence it could be interbreeding was prevented. The great tree frog gives a short shrill sound, very loud, lasting for about 3 seconds, while the pine wood tree frog makes a call that consists of a series of short dots and dashes, right. So this is one typical example of the ethological or sexual isolation. Another is with respect to pheromones. Okay, pheromones, they are chemicals which are released to the external environment by the insects to communicate with the members of the same species. Okay, not only insects, even mammals you can see. And these pheromones, there are 6 pheromones, and these moths, what do you call the so the moths, they release sex pheromones for attracting the opposite sex, right. And it is very species specific. The pheromones are very species specific. Clear? The next kind of isolation is morphological or mechanical isolation, and this type of isolation comes about because of different sizes or shapes of external genital organs, making copulation and sperm transfer impossible. Okay, and typical example is that of two toad species, that is Bufo quercicus, it's a minute octod, and Bufo valliceps, that is a Gulf Coast toad. Okay, here you can see that the female of the octod, the minute octod, is very small, attaining a maximum length of 1.25 inches, and some adult Gulf Coast toad, they are 2.25 inches in length. So here size alone prevents interbreeding. The male octod is too small to grasp a female Gulf Coast toad, while Gulf Coast toad are so large that they are more likely to eat the female octods than to try and breed with them. So this kind of a barrier, it can actually prevent interbreeding. Okay, even there is an example from Drosophila here. It was based on the study conducted by entomologist Dufour in 1844, and he states that there must be a very exact correspondence between the morphology of male and female genitalia to permit copulation. He compared the female genital organs to a lock and the male into the male of the same with the key. So for example, in the case of Drosophila, the interspecific crosses, that is crosses between members of different species, it resulted in injury or even death of the participants because of the incompatibility of sex organs. So this one, it is from the insect world. Okay. The next kind of prezygotic isolation is physiological or genetic incompatibility isolation. Okay. So here what happens is mating may take place, but gametes are prevented from fertilization. So here it is very clearly given, it is genetic incompatibility. Sorry, it is not genetic, it is gametic. Okay, it is gametic incompatibility, that is gametes are not compatible. Okay, because of gamete incompatibility, even after mating happens, because of gametic incompatibility, fertilization may not take place. Okay, so this actually prevents the formation of zygotes. Typical example is from that of the Drosophila. The study was conducted by Patterson in 1946, and it was shown that certain species of Drosophila mating among the members of different species is not possible. This is because the vagina swells up immediately after copulation due to rapid secretion of the fluid, and the swelling may last for a few days if copulation is interspecific between the members of different species. Sometimes the secretion inside the vagina solidifies and blocks the passage of eggs, making the female sterile forever. But if copulation occurs between members of the same species, that is intraspecific copulation, in that case what happens is the swelling may last only for a few hours, and after which the vagina returns to its normal condition. And if the gametes are killed inside the genital tract, the isolation may also be called as gametic mortality isolation. Okay, so here what happens in the case of Drosophila, only the interspecific mating can result can give like harmful effects on the vagina. Okay, so that can result in certain reactions occurring in the female Drosophila. Okay, can be referred as insemination reaction. Okay, right. Now next, the last one is cytological isolation. Cytological isolation, here fertilization cannot take place, and it is due to the differences in chromosome number between two species. One example is from again from the frogs, the Rana species. Two frogs, Rana clamitans and Rana catasbiana, they do not hybridize mainly because of cytological block to fertilization. However, you can see here that the chromosome of the two species are incompatible. Okay, and hence fertilization cannot occur even if they are brought in contact with one another. Clear? So cytological isolation, these experiments have been conducted by many scientists. Among even these two are closely related, but even between entirely different species, such kind of experiments have been conducted, and it has been seen that wherever there is a chromosome number incompatibility, the hybrid zygote formed will not survive for long. Okay, so these are the different kinds of prezygotic isolation. Here you can see this one is geographic isolation. So two subpopulation of the same species being separated by a geographical barrier, and here it is mountain ranges, right? Ecological isolation or habitat isolation, where the two subpopulations of the same species, they survive in the same territory but in different habitats. One here in this particular example, this is ground dwelling, and this is found on the tree branches. Okay, temporal isolation, that is, you can see in the case of plants, the breeding happens when it flowers, isn't it? So the flowering season is associated with the breeding time, and here you can see the flowering season is of these two plants, they are not at the same time. Here it is during the fall or the winter time, and here it is during the springtime or the summer time. Okay, and another example you can see here is behavioral isolation. Okay, you can see two examples. One is with respect to you can see here in 1 minute. Okay, let me just move that. So here you can see two different populations of similar kind of birds, right? They are making a call. Okay, here this actually gives the notation. Okay, and here it is widely spaced while it is closely spaced, and these two birds, it cannot interbreed because it cannot respond in a positive way to the mating call. Similarly, this is with respect to the fireflies. Here you can see 1, 2, 3, 4, 5, 6. Okay, 6 species of fireflies are given. Fireflies, they communicate for mating by giving away flashing lights. Okay, yeah. So these flashing lights, actually they are species specific. We can see the first population, the first species, you can see here 1 2 3 and then a pause, 1 2 3 again a pause. So that is the kind of a pattern of flashing light. What about the species B? It is continuous, isn't it, with spaced lighting flash. Now in species C, how it is? We can see continuous flash and then a long pause. So likewise you can see each species is distinct, and this particular species A, when the light is given, obviously the female responds to it in a positive way, while the members from other species will not respond to it. Okay, so it is so specific. That is kind of a behavioral isolation. Another is mechanical isolation. Okay, you can see the two different kinds, and here you have as the example from the aquatic what you call crustaceans. Okay, and here it is gametic isolation, or it is referred as a cytological isolation. Okay, so these are a few examples of prezygotic isolations. I hope it is clear. Okay, now we'll pass on to the postzygotic isolation. Postzygotic isolation is, once the postzygotic isolation barriers, it will never prevent the formation of zygote, but it will reduce the viability or fertility of the hybrids formed. And there are three ways by which postzygotic isolation works. First is hybrid inviability isolation, then second is hybrid sterility isolation, the third is hybrid breakdown isolation, or F2 breakdown. Okay, hybrid inviability isolation, here in this type, hybrids are formed by interspecific crosses, that is, members of two different species may interbreed and give rise to hybrids. Okay, but the individuals developed from such hybrid zygotes, they die before reaching sexual maturity. So death or inviability may occur at any stage in the life cycle. It could be immediately after fertilization, or in the embryonic period, or after birth, but before sexual maturity is attained. Okay, when the mortality happens immediately after fertilization, such kind of process is known as zygote mortality isolation. Okay, zygotic mortality isolation. Okay, that is soon after the formation of zygote, soon after fertilization, the zygote undergoes death. Okay, best example is from hybrids of bullfrog and gopher frog. Okay, bullfrog hybrids between bullfrog and gopher frog, they pass successively through early stages of development but die before reaching the tadpole stage. Even though these very rarely interbreed, but interbreeding happens occasionally, but what happens, they may not reach the tadpole stage. Okay, because of zygotic mortality isolation. Fine. Next is hybrid sterility isolation. Here certain interspecific crosses may result in the production of sterile hybrids. They may live to sexual maturity, but they cannot produce functional gametes. Okay, they are sterile, so they can even though they can reach the sexual maturity age, but because they are sterile, they cannot produce functional gametes due to chromosomal incompatibilities. Okay. The best example is provided by, with the hybrid mules, hybrids of horse and donkey. Okay, these mules, they can reach sexual maturity, but they are not viable, so they cannot interbreed or give rise to the next generation. Okay. Now the third one is hybrid breakdown isolation, or F2 breakdown. In this type of isolation, the F1 hybrids are highly or at least partly fertile. Okay, F1 hybrids means two parents intercrossing giving hybrids. Okay, the two parents will be from different species. Okay, these two interbreed giving rise to fertile individuals. Okay, hybrids are formed, and these hybrids are fertile. But what happens? The hybrids may give rise to weak, abnormal or sterile progeny in the next generation, in the second generation. Okay, so this phenomenon is known as F2 breakdown. This is why it is known as a F2 breakdown, because F1 hybrids are fertile, but reaching F2, it becomes weak, abnormal or sterile. Okay, example is from the hybrids of Drosophila pseudo-obscura and Drosophila persimilis. Okay, so F1 hybrid females formed from a cross between these two Drosophila species are vigorous and deposit as many eggs as non-hybrid females, a normal female lays. Okay, yet when these females are back crossed to males of either parental species, individuals of both sexes in the offspring produced will be weak and sterile. Okay, I hope it is clear. Okay, that is the F2 breakdown. At the F2 generation, what happens is the individuals produced will be weak and sterile. But F1, it will be almost just like a normal one. F1 hybrid will be just like a normal individual. Okay. So this is all about the isolation and isolating mechanisms. Okay, fine. Thank you.